and say like, okay, you're almost there. Okay.
Yep. Okay, so now the recording has started. I'm going to go ahead and mute myself. Just wait like 3 seconds and then you can begin your talk.
Hi, thank you so much for this amazing opportunity to speak with all of you. My name is Kanaka Rajan. I am an associate professor at Harvard Medical School and a founding faculty of the Kempner Institute of Natural and Artificial Intelligence. I have been working on computational and theoretical models that are constrained by neuroscience data. And in doing so, we try to understand how the real brain inside of animals and humans learns, remembers, and decides, and is capable of doing the incredible repertoire of tasks and behaviors and cognitive abilities that we see. So one of the amazing moments in history right now is the fact that we have incredible amounts of data from the electrical activity of many neurons in the brains of different animals. And so this is a graph that shows you where we really are. So we're in this era of unprecedented explosion in neurotechnologies that let us able to collect this kind of data. And so you can see that as time goes on, our ability to monitor more and more of the brains of animals and in some cases humans, as we're able to do different tasks and behaviors, has kind of grown exponentially. So one of the things that computational modeling affords us is the ability to make sense of these data sets. And then theorists like me come in to try and make sense of these types of data to extract essential principles from them. And then we write down theories to put them in perspective. So one of the most common approaches that people like me take when we're faced with large-scale data, such as hundreds of or thousands of neurons over many minutes or many seconds, is to do what is known as dimensionality reduction. And so what that actually means is to take the outputs of experimental preparations, such as these electrical activity traces as a function of time, and perform this mathematical operation that lets us essentially transform the entire population's dynamics into a new coordinate frame, such that the entire population is represented by this kind of trajectory that changes over time. So one of the observations that many labs have made and many data analysis approaches have yielded is that when animals and humans perform various tasks and behaviors, then the neural activity across many neurons in the brain traverses trajectories that are typically smoother or lower dimensional than the total number of dimensions that is available to it. So this is an example of what is known as a neural manifold. And so these lower dimensional subspaces can be defined by looking at the pattern of covariation of neuronal outputs that are collected experimentally in various labs. So one of the things that this view misses. So first of all, I should say that this view of looking at population activity in terms of manifolds is extremely powerful, and we've done some incredible work by looking at rotations in these types of subspaces, for example. But one of the things that this output-centric view misses is the following question, which is, what are the inputs that shape manifolds and where they're coming from? So what neurons are telling you, what you're able to measure from neurons, are really the what it is that they're doing. Inputs really are the path to mechanism, which is the statement of why and how a certain pattern of neural dynamics may emerge. And so you can imagine these inputs could be external inputs that come into neural networks in the brain, like the schematic you see here. But one of the specific kinds of inputs I'm going to be talking about is interactions within and across brain areas. So in the schematic that you see here, multi-region interactions are a form of inputs that shape neural manifolds. And so there's a few things that the output-centric view that we currently have of looking at manifolds in the state space of outputs that are collected experimentally is inputs to each neuron from within and across brain areas. So if I'm a neuron sitting in area A, You want to ask how much of my activity comes from interactions that I have as a neuron in area A with other neuron A areas, and other neurons in area A, and how much of my activity comes from interactions area A has with area B and area C. An output-based view does not easily let us capture that. There's a couple of things that are still inaccessible from current methods of analyses or from measurements, and that is the directionality of interactions and effect of common inputs. So imagine for a second that area A were the common input area that drives both area B and area C, or are area B and area C areas that are upstream driving area A. Both these scenarios that I've just sketched out for you look exactly the same from the pattern of covariation in neuronal outputs. The neurons will covary with exactly the same sort of profile, independent of whether they were connected one way or another, or if all three areas are connected reciprocally, like I've drawn in the schematic. And finally, current analysis methods and current methods of measurements struggle to scale to more than two areas, although the field has made tremendous progress in this area. And the approach I'm going to be telling you about today tackles these shortcomings sort of head on. And so how do we get at mechanisms that are inaccessible from measurements, right? So one of the approaches that my lab really takes a lot, and it's built on work from other labs also, is that we build data constrained multi-region models. And so when I say data constrained, I mean that we build these models that are trained directly to match neuronal and, or in some cases, both behavioral data as well. And so these types of data sets can come from either synthetic or idealized data, like I've shown you in this drawing here as ground truth data. It can also come from small nervous systems in which we can practically sample the whole brain, like in fruit flies or larval zebrafish, or from larger mammalian nervous systems like mouse, macaque, and humans. And then once we build these models, we analyze them using new methods and then some of the same methods as those used on data. And finally, our goal is to infer mechanisms that are intractable from just measurements. So can we go or extrapolate beyond the details of a single experiment to extract common operating principles? And that's the whole program here. So now one of the things you might notice when you look at this circle that represents all these different nervous systems that provide us data is that behavioral complexity grows from system to system, at least to our eyes, right? Like the behavior of humans and macaques looks more complex than the behavior of fruit flies and larval zebrafish to our eyes. But also our access to how much of their brains we can sample or measure directly drops precipitously as you go around the circle. So one of the things that we can do is to actually mimic the literal sampling ratio. But instead, what I want to convince you of today is that consistent mechanistic inferences, that is inference of consistent mechanisms, are still possible with qualitatively similar amounts of data. So by that, I mean, if you have an order few hundred neurons simultaneously recorded or even in parallel recorded from these different data sets from 3 to 8 regions or something, we can still make progress and extract consistent mechanistic inferences that are intractable from measurements. So how do we do this? So in order to do this, so first of all, for our younger colleagues, I want to say that the class of models that I'm going to be talking about today are recurrent neural network models, or RNNs for short. And that's just the statement of saying that each network is made up of model neurons or units that are neuron-like. So they're mathematical abstractions. And rather than excitation flowing like a bucket brigade from one active neuron to the other unidirectionally, instead, we have connections that go forwards and backwards. So we have recurrent connections that define these networks. Now, these networks, the single module RNN that you see on the left has been studied extensively before. But what we've now done is to connect many of these RNNs together with perhaps slightly less dense connections, as I've shown you here. And so this is how we build a multi-region RNN. We just take many of them together, as many as the areas in the experimental data set, and we wire them together. My PhD work was based on properties of these types of recurrent neural networks. So that's the first thing we do. The second thing we do is to train the activity of these units to match data directly. And what I mean by that is to take the activity of each neuron-like unit in this model, which I've symbolized here in this red trace here, and also schematized as ZI. And I want to train it at every time step to match a target function that is derived from, let's say, calcium imaging data, and that is symbolized here as Fi of t. For every neuron I, there's a target function. And then at every time step, the entire matrix of connectivity changes until the learning error is minimized, or the instantaneous difference between these two traces is minimized. So this is by no means a realistic plasticity mechanism, but it's a trick to get these models in the right initial condition. And then we typically build models that are as large as the experimental data set. And so I'm going to show you a very large example today. However, we've also built large networks and done subsampling of it. And I can tell you more about it as we go on in the talk. So what is this exercise bias, right? So we connect more than one RNN together to make a multi-region RNN, and then we train the activity of each unit in this model to match data directly. So what we gain from this exercise is, well, once you stop training these networks, they produce dynamics generatively. They produce neural-like dynamics that are realistic and stable. Now, that by itself is not a huge surprise for the theorists in the crowd, because these models are very powerful and we have trained them to produce this kind of activity, so they will. And they do so very, very successfully. But in addition, they can help us infer from the trained model directed interactions. So what do I mean by that, right? So I'm back to the same picture I showed you 2 slides ago, but now there's another row to it. So here I'm showing you the pairwise interaction between every neuron and every other neuron in this model, right? So in the columns are the sources and in the rows are the targets. And each square here is filled with how strong and what type, is it an excitatory or an inhibitory connection that these neurons are paired with each other with. So this one succinct mathematical object, this matrix, tells you the directionality, the magnitude, and the type of connections in this model. And it turns out that these models are a simplification of the real biology, but they do pretty well in capturing the richness of the behaviors that can emerge from these models. But when you start to build a multi-region RNN by connecting many of these modules together, this matrix, it's still a matrix, but now it starts to have interesting properties. So for example, if we did this exercise cleverly and built it so that the inter-area projections are less dense, or in other words, sparser than the within-area connections, then this matrix will have denser connections in the square here, A, B, and C, that you see along the principal diagonal. And it'll have slightly less dense connections in these other squares. But in addition, this matrix allows us to separate connections within area A in this blue square, and then these little ombre color squares tells you connectivity from B to A and connectivity from C to A. So the same mathematical object, this one matrix, can help us disentangle within and across area connectivity based on this matrix. So that's like the second big get from training these types of models. The third get comes from the observation that if you take this matrix of connectivity and you multiply it with the activity that you found, either you recorded this experimentally or you fit it from the model, you get this idea of currents. And you get consistent within and between area currents just based on how we do this dot product, right? So if you just take the entries inside the submatrix and compute the current, you'll get the current within area A. And if you do this multiplication with different submatrices, you get these different subcurrents. These quantities, these last two things I told you, the inference of brain-wide interactions and then the consequent extraction of these currents, are impossible to track experimentally. And so this is the real get from building these types of multi-region models that are constrained directly by data. So what does that look like, right? So let me show you what this looks like in practice. So now to do that, I'm going to extract, the ground truth model. So I'm going to just build myself a fake animal from which I'm going to construct some surrogate or idealized neural-like data. So this is a thought exercise, if you will. So let's say we have this fake animal that only had 3 brain areas in it. And I'm mimicking that scenario by constructing a toy model in which there's an RNN-A in blue, RNN-B in yellow, RNN-C in red. And for argument's sake, I'm constructing the inter-area projections to be 1%. So they are sparser. Now imagine that an experimental colleague collected data from these 3 regions, and that's the output-based view that I'm showing you here. So I'm only picking 1 neuron each from these areas. So imagine that region A has activity that looks like this, neuronal activity or firing rate as a function of time. Let's imagine that I've changed the parameters of this toy model so that our eyes have something to look at. So region B, rather than making something completely random, makes this kind of temporally sparse bump. And imagine that region C makes a fixed point that goes to another fixed point at some point here. So what we can now do is take a wholly different unstructured network, which I'm calling the model RNN here, and I'm going to fit it to the data produced by this toy model. And when I do that, now keep in mind, I don't tell the model RNN how many areas to expect. I'm just throwing the whole grab bag into this fit. And once I do that, I can extract from this fit this kind of matrix of connectivity that I just told you before, or this directed interactions matrix. Now, with this matrix, I'm able to decompose the activity that was recorded in each of these regions, but just in this illustration here, activity recorded in region A into these components, or in fact, the inputs that are responsible for producing the outputs that you've collected experimentally. So doing this decomposition using this matrix gives us an A to A current that looks eerily like the region A's output, not surprising. But in addition, it gives us a B to A current that has the bump that is reminiscent of region B's activity, but is invisible in the output-based view of region A. And it also gives you a C to A current that looks exactly like the output you would have recorded in region C by having this like fixed point transition to this other fixed point. The sum of these 3 still trivially give you the output that you would have recorded. But if we didn't fit this model RNN to this toy model or the ground truth data, you wouldn't have this matrix of connectivity. If you didn't have this matrix, you couldn't do this decomposition in the first place. So this method really allows us to decompose recorded activity into the source currents within and across areas that are responsible for producing it. And consequently, we call this method current-based decomposition of population activity, or CURBD for short. So we've applied this technique called CURBD to data from all of these systems. And what we're trying to do with this method is really in vivo tract tracing from many different data sets. So we've applied it to data from small nervous systems as well as large nervous systems. But really what CURBD can help us figure out is whether a recording is from a single area or multiple distinct areas, whether there's a dynamical difference between areas, whether there's a structural difference between areas, and finally, if there's a difference in the types of inputs each area receives. And the code for this, as well as the data set, are available open source on my lab's GitHub. But we've been applying this to many different data sets, as I'd mentioned before. But I would like to show you what happens if we apply this method to a nervous system in which we have no sampling issues. So a small nervous system, such as the larval zebrafish system, felt like a nice via media point. So now I've shown you what it did on ground truth data. I want to take you in a more experimental direction. And so the data that I'm showing you here come from larval zebrafish, and you see a cartoon here. And the areas in the larval zebrafish brain are kind of pseudo-colored. And this is what the activity of each neuron in the zebrafish brain look like. And the fish are only head fixed. Now, these data come from our long-term collaborator, Carl Deiseroth, and they were collected by various postdocs and trainees in his lab, and we're really grateful that they collaborate with us on building these models. And so a head-fixed larval zebrafish, and they express nuclear localized calcium indicator. So every neuron glows with a certain time constant. We're also able to monitor the tail movements of these fish. So you can see that in the blue dots, and it's continuous representation in this blue trace here. And we're really able to collect these kinds of brain-wide calcium activity traces over really very long times. So you can see the time scale, the scale bar over here or over here is over several minutes. And so these activity are large-scale activity. There's no task. The animal there isn't a start time. There are no trial structures. This is an animal that is just spontaneously behaving. It does wiggle its tail, but it's in the absence of any explicit reward. And so this is what the activity looks like in pseudo color from one region here that I'm showing you here, telencephalon. And on the right-hand side of this, I'm showing you what happens if we did dimensionality reduction, like I had alluded to in my very first slide. In fact, we're able to collect between 10 and 60,000 neurons per animal over a function of very long periods of time. And there are only 100,000 neurons in the brain of this animal. So these are really very large-scale data sets. In fact, this is what data from one animal looks like. And I'm showing you all of the regions that we're able to monitor, and we're able to monitor the tail movements at the same time over a period of time. So the typical way to make sense of this data is to really do dimensionality reduction, and you can see the results of plotting those low dimensional manifolds on the right-hand column of this slide. So you can see these do make kind of smooth excursions in the state space. And the traditional way of looking at this would be to just correlate these excursions with the timing of the tail movements and try to make sense of these data. But we wanted to ask the following question, which is, and also if you wanted to look at something like the correlation matrix or the pattern of covariation of the outputs that are collected experimentally, there is no obvious structure you see in this covariance, right? Like you can see that this is, it looks like it's just it's information rich, but there is no obvious structure that pops out even within each area. And you certainly can't tell if there's information flow across areas. So the thing that we decided to do, admittedly inelegantly now, is to say, well, what if we modeled the whole brain as a multi-region RNN? And so that's what the schematic is showing you here. On top is the experimental data set. And here's a schematic of how we're conceptualizing this multi-region RNN. And exactly as I showed you before, every neuron has a target function that comes from the experimental data directly. And here are all the regions that we expect to find, because we're not telling the RNN how many areas to expect, but we do have the identities from the neuronal data set itself. So that's one thing we do. The other thing is the observation that this is several minutes worth of data, right? There's no obvious timestamps to it. This is several minutes. And so we didn't want to make the assumption that there's one matrix of directed interactions that controls the entire dynamical system. So what we did to make sense of longitudinal data or long-term recordings like this was to take the data set and chunk it into epochs that you see here. And so here we just decided on 6-minute epochs, and the details are in the preprint as to how we picked the length of this epoch. And what we end up doing is initialize the ANN to the final trained condition from the previous epoch. So in some sense, there's continuity in the training, and we're calling this approach chaining. So instead of getting this one matrix of directed interactions that we expect, we get a whole chain of them. So rather than getting a single matrix, we get a whole tensor. And we use a very similar learning rule to extract this, like I had already shown you before, except we do this process epoch by epoch. And the initial condition for the training of each of these ANNs or RNNs is the finally trained matrix extracted from the previous epoch. And this is the same learning rule. It's exactly the same thing that you saw before. So what does the entire pipeline look like, right? So in order to make sense of these, we developed this pipeline. And so we wanted to make sense of this, and that's what you see here. So this is the multi-region RNN that's fit to data. And underneath, for the statisticians amongst you, I'm putting in the dimensionality of each of these objects that we extract. And this is showing you a schematic of the chaining over time. And the result of that is this tensor of connectivity, where you can think of it as slices of bread. And then we run the same exact current-based decomposition or curved on these matrices serially by multiplying the entries in this matrix as a dot product with the activity that is collected experimentally. But rather than getting one curved output, like I showed you in the toy model, we instead get a tensor that looks like this, right? So there's a pattern of sources, times, and targets. So it's a 3D object because it's a 3D object that we're using to multiply. Now, to make sense of this tensor, we use a method called tensor decomposition. It's also a method of dimensionality reduction that gives you a set of low-dimensional modes that look like this. So each of these is a low-dimensional mode that has a pattern of sources, a pattern of targets in green, and a time series. Now, since these are inferred from a model fit to data from a single fish and can be recomposed, we call these compositional modes. So this is what each of these modes look like. So each mode has now 3 components. There's one component that corresponds to targets or weights. There's another component that matches source weights. And then there's a time course that can take any shape, but in this case, it looks like a ramp. Now, we can extract many of these modes, but we also can use these as loading weights to put the loading and the engagement into the atlas of the brain of the fish. So we can really use this information to repopulate the brain of the fish in these kinds of visualizations. So I can tell you that each mode corresponded to X% of engagement of these different areas. So you see in this brain of the fish, you see what kinds of engagement across areas was involved. And so what do these modes really look like? And here's an example. So here's compositional modes extracted from RNNs that are fit to data from a single fish. So there's a mode that has this ramp that you just saw before, but in addition, there are these modes that have these different sorts of waveforms. I'm showing you 5, and you can extract many more than this, but eventually they start looking like copies, and that's how you know you have sufficient number of these modes. So this is what they look like. There's some variation across the shapes and the time courses and the spatial engagement across the brains in these modes. However, what we were surprised to find is that there's a ramp-like mode in all of these fish. We didn't expect there to be. Remember, there's no trial, there's no task that these animals are doing. They are nonetheless head fixed. And remarkably, we see this consistency in both the time course, in that there's a ramp in all of the fish, but all of these also correspond to very similar spatial patterns of engagement across areas. So there's a ramp-like mode from all of the fish. In fact, we see that there's a whole lot of shared or conserved compositional modes across individuals. So we find that there's a ramp-like mode that I just showed you, but in addition, there's this thing that we're calling an oscillating mode that has this particular time course. There's a fast decaying mode, and then there's a slow decaying mode in all of these. And so what we are really seeing is that there's variation across the modes, but there are compositional modes that are conserved across different fish. And this is kind of getting at a solution to a long-standing problem in computational neuroscience. Namely, how do we trust our models? If I build a model and another lab builds a model, how do we know that they're on the same units to be able to compare? And this consistency gives us hope. It's because there's two sources of noise in it. There's the initialization in these matrices and these models, but in addition, these data come from entirely different individuals. So the fact that we see this kind of conserved motifs across different fish is actually heartening, meaning that our idea of fitting these models is actually on the right track. So is this all there is in the brains of these fish? Is the whole brain-wide activity just made up of these Lego blocks of compositional modes? No, we also find compositional modes that are unique or specific to individuals. And in fact, those modes capture a quarter of the variability of the total activity, and they seem to be more correlated with the total brain activity than anything else. And so that's what that looks like. The orange trace is the sum of the brain-wide activity of all of the neurons. And we see this compositional mode that was left over. It's a residual compositional mode that changes from individual to individual. And so you can see a few of those examples, and they seem to be more correlated with brain-wide activity than it is to the common motifs. So yes, so we have so far inferred such modes or compositional modes consistently. Question is, can we really perturb them experimentally? If we cannot perturb them experimentally, we cannot call them compositional anymore. And so we wanted to try a couple of things, and one is a behavioral manipulation. And so this is the compositional modes from the ramp-like mode from a spontaneously behaving fish that you already saw before. But here's the experimental paradigm in which the same head-fixed fish are shocked with 1.3 milliamp shocks for about 15 or 20 minutes. Now, in our 2019 Cell paper, we had shown the mechanisms inside the brain of these fish that lead to this kind of behavioral transition, where fish go from actively coping in the initial parts of the stress to passive coping, where they respond by volitionally turning off their movements and going into a state of almost passive coping, of learned helplessness, as the stress is perceived to be persistent and inescapable. So we have data collected brain-wide during the stress paradigm. And so what we see in the shocked fish is that these ramps that we saw in the spontaneously behaving fish do change somewhat, but really strikingly, the spatial pattern of engagement of various neurons, of various areas in the brains of these fish are changed relative to the spontaneously behaving fish. The other aspect of these data that I'm showing you that give us hope is the fact that these two areas that you see here are the habenula nuclei in the brains of these fish, and hyperactivation of the habenula, either optogenetically or behaviorally or idiopathically, is known to be a predictor or related to conditions such as learned helplessness, and has even been associated with depression in mammalian systems. So this was an observation that came from the model without us making any assumptions about areal identity or their specific functional roles. So the answer is yes, we can manipulate the model inferred compositional modes behaviorally. We can also do a second manipulation in which we can apply ketamine to these fish. So this is the speed at which the fish make movements as a function of time. In blue is the result of the active and the passive coping transition. And you see the shocked fish really do cease their movements about few minutes into the challenge. But if you apply ketamine, then the onset point of passive coping, which is manifested in the cessation of movements or tail movements in the presence of stress, is dilated relative to the shocked fish in which ketamine has not been applied. So when we look at compositional modes from this scenario, we were hoping that somehow the fish treated with ketamine will have a reversal in that they will start to look like spontaneously behaving fish, but the answer is a little bit more complicated. So a few things we observe is that these ramps that we see in the spontaneously behaving fish or in the shocked fish are still different in the ketamine fish. And they're consistent across individuals in the spatial pattern of engagement. But the habenular hyperactivation that you saw in the shocked fish, like this is a particularly striking example, reverts back to much closer to the control fish condition. So it's possible that there's a habenular centric mechanism that is responsible for both the passive coping transition, as well as why the passive coping transition appears to be dilated in fish that are treated with ketamine. And in the preprint that is about to hit bioRxiv any day, we've done some statistical controls to assure ourselves that what we're seeing isn't an artifact of one of the methods we've used, but in fact is a real measurable effect of these compositional modes. And so how am I seeing this picture,
Hi,
right?
Kanaka.
So here's yeah.
Hi, sorry, just a quick question. Do you know how many slides you have left? Because we're at the 35-minute mark. Oh, this is the last slide?
Yeah, this and acknowledgement.
Okay, perfect.
So should I go back here? What am I doing?
You can just start. No, I waited until you finished this slide, so you can just start on the next slide.
Perfect.
Awesome. Thank you, Kanaka. You can start whenever.
Should I start now?
Yep. You might want to switch the slide first, or
you may. Yeah. Okay. So at the end of this, I want to say, I want to leave you all with a big picture reconceptualization of how we're seeing this kind of modularity at the neural dynamics level that I've shown you with these compositional modes. So we're seeing these compositional modes really as a grammar for the dynamical coordination of behavioral states and transitions. And so one way of looking at behavioral transitions is to look just at the behavior and seeing that every time, let's say the fish makes a leftward turn, you can call it a behavioral syllable. Every time it stops moving, you can call it a different behavioral syllable. And those are driven by these internal processes that we don't know exactly how to measure or conceptualize. So one way of thinking about this is this discretized view of neural modularity in which there's an area A that works like a switch, in that every time area A is active, the fish makes a leftward turn. Every time area B is active, then the fish goes, kind of tilting towards the right and nowhere else. This is not inconsistent with some behaviors such as aggression or parenting. However, when we're thinking about sort of neurotypical adaptive behavior or spontaneous behavior in the absence of any tasks, the view that I presented to you today seems to be much more consistent with experimental data. And that is this view of looking at currents in which these compositional modes are the ones that are responsible for mediating the behavioral states and the transitions between them. And they are almost agnostic of areal identity. They are only responsible for coactive ensembles of neurons. So really, we're proposing compositional modes as state trackers or transitions between behavioral syllables. So in my lab, we're really proposing this kind of reconceptualization of the grammar for the dynamical coordination of behavioral states and transitions, thereby merging the two levels of modularity that our field struggles with: behavioral modularity in terms of behavioral syllables and neural modularity in terms of area-specific engagement versus this kind of compositional, squishier way of thinking about neural modularity. And with that, I just want to leave you with just a few, just the summary slide that tells you what I told you about today, which is the power of these multi-region RNNs. I explained to you this curved technique that my lab has developed, and using the curved technique, how we're conceptualizing the grammar of behavioral transitions in the brain. And I would like to thank my lab, key collaborators, particularly the Dijsrath Lab, for their help and working with us over a long period of time, and funding sources for their faith in our ideas. Thank you.